more or less, actually it's not really in order, so, what do you guys think about this first one? And can you guys see the font in the back? Just forget about you guys.
Looks like
You can't see it at all? Oh. That's a very font. Yes, sir. Okay, so we're talking about chlamydia, this question. The two different forms. Which one is infectious? Elementary, vowel, infectious elementary. And is that bigger or smaller than others? Smaller, yeah, 300-400 nanometers. Very good. What do you think about this one? Not trichomonas. Trichomonas.
That's what we said.
Trichomonas, and the other one was pneumoniae. Yes, very good. Kinds of cell types are susceptible. That's a little redundant. What cell types are susceptible to infection by trichomonas? To answer that question, it kind of helps to think about the different disease presentations. So what's one of those three disease presentations that we talked about? The urogenital one, yeah. So we're talking about what kind of cells if we're talking about urogenital tract infections. Epithelial cells, yeah, exactly. So urogenital epithelial cells.
Is this like SUK or like DUK or something?
Yes, but your plot spoiling one of my subsequent questions, but yes,
Whatever.
yeah. Okay, so we have the urogenital tract infections where those susceptible cells are the like the epithelial cells and the rest connects to the respiratory tract, urogenital tract. And then chlamydia trachomatis can also cause what other types of infections? Right, so then we can infect those cells as well and cause trachoma. And then LGD, now we're talking about, man, I think I just blew all these questions, but we're talking about a much more invasive form of chlamydia that can happen, urogenital form. All right, Cerevars associated with trachoma, eye infection. A, B, A, C. Yep. And then urogenital tract disease, D through K, and then LGB. Yes, exactly. L1, L2. Very good. How is LGB different than the STI infections we associate with D through K? It's more severe. Yeah, chronic formed granulomas, some of those. Invasive. You can get suppurative draining lymphatics, that. So, yeah. And do you see a lot of LGB in the United States? Not very much, no. Test of choice for chlamydia trachomatis? PCR, yeah. Why would we prefer to use PCR for chlamydia? Yeah, it's intracellular. What do we know kind of as a generalization about intracellular bacteria? Harder to culture generally, yeah. So PCR is very, very sensitive. We do lots of high throughput testing on those big platforms. Okay, we talked about atypical pneumonia. Walking pneumonia, what does that mean?
Mild onset, high fever.
Walking pneumonia means you feel
fine.
functional. Yeah, so it's usually kind of associated with you going out and being among other people and not staying home so you can transmit the disease. But it looks pretty bad.
You used the term on Monday. It was the difference between asymptomatic and you used another term like
Subclinical?
Subclinical versus asymptomatic.
I think they're pretty much the same thing. So subclinical means you are not really presenting. And that can be true of a number of things. You have the organism in you, but you're not symptomatic.
But subclinical means you could present later versus asymptomatic means you never will?
No, asymptomatic doesn't mean you never will. So you can be asymptomatic now, and then tomorrow concentration enough bacteremia, viremia, whatever you're cooking up to be symptomatic. Or if it's immunopathology, then, you know, something related to immunopathology causes the symptoms. Yeah. We talked about some bacterial structures and some things that are distinctive about these different organisms. No cell wall and no peptidoprotein. That has implications for antibiotics. Anything you want to remember. Antibiotic treatments of mycoplasma. The two species that we talked about in this genus, Pneumoniae and genitalium. Yeah, mycoplasma genitalium. Yeah. With pneumonia, how is that transmitted? Respiratory droplets, person to person. Before I move on to this question real quick, what kind of population do we sort of associate with mycoplasma pneumoniae? Young adults crowded together in close contact. Okay, rickettsial stuff. Cell types susceptible, also redundant, susceptible to rickettsial pathogens. Epithelial. What about if you get a rash and you have like petechiae? What's that indicative of? Vascular damage, so endothelial cells in blood vessels. Rickettsial diseases have a tropism for endothelial cells. Okay, RMSF. You guys remember this is a hard one, what bacterial species causes Rocky Mountain spotted fever? Yeah, it's the redundant one, Rickettsia rickettsia. What arthropod vector is this bacteria? The American dog tick, yeah. You know the scientific name of the American dog tick? Yes. Ermacenta variabilis, that's like the east coast one, I think the western states have Ermacenta andersoniana. Yeah. And then the region, we're talking mostly Rocky Mountain states, yeah? No, not Rocky Mountain states. What states have the highest number of cases of RMSF in the United States? They're more eastern, right? So we've got
North Carolina, California.
not California. There's an acronym I remember, it's not super helpful, but NATM, so North Carolina, Alabama, Tennessee, Arkansas, and Missouri. Or MADA, whatever acronym you want to use for that. We'll go through all of these guys. So when we're talking about epidemic or classic typhus, what's the bacterial species that causes that? By the way, the name of that book I was trying to think of yesterday or on Monday was Rats, Lice, and History, and Zinsser. Zinsser.
I said he had Zimmer yesterday, I was like, he's right before I
Probably did, yeah, writing musical scores.
Did he make that before or after Guns,
Did he, Guns, Germs, and Steel?
Did he write that before or after?
That was Jared Diamond.
Yeah, but he stole the name, he stole the.
Oh, I don't know. I don't know what your Rats, Lice, and History was published, but Guns, Germs, and Steel is different too. What's that? Okay, sorry, let's try and squeeze this one out if we can. Crowsapra, yes, and then since it's epidemic, that kind of gives you a little bit of a hint as to what vectors it. Sporin, yes, lice, and then what regions are affected? Man, it's really the morning for you guys. Okay. Worldwide, because that's where the lice are, but generally associated with what kind of conditions? Unsanitary conditions. Never said that. Good job. Yes, unsanitary and crowded. All right, endemic urine typhus. Which bacteria causes it? Yeah, Rickettsia typhi. Yeah, no, it's coming out of the feces, and you rub in the feces, that's how it's transmitted. And then region for this, tropics in Texas, yeah, we get cases here in Texas. Is the presentation more severe than classic epidemic or less severe? Less severe. Okay, and then finally, last but not least, scrub typhus. Organochet, Tsukumoshi. One word. And then the arthropod vector for Tsukumoshi. The mite bite, yes. Region. East Asia, large swaths of East Asia. Well, deep in Asia as well. Okay. Coxiella burnetii, a couple questions about this. When we're talking about its ability to survive out in the environment, which one of these forms, what do those stand for? LCV is large cell variant, small cell variant. And which one is the one that is more rugged and able to survive? Small, yes, kind of a rule of thumb in nature. Primary reservoir for burnetii. Livestock, yeah, livestock, goats, cows, whatever. How do we get this disease caused by Coxiella burnetii called Q fever? We can inhale it from being around livestock. What's another route of transmission? Unpasteurized milk, drinking that. Another route of transmission? Ticks. Yeah, good. Why is Q fever so difficult to diagnose? The presentation is difficult, it's rare. You get like 200 cases a year in the United States, so we don't see it very often. CDC would probably have to get involved. Is it a biological threat agent? It is. Okay, last one. 4 questions packed into one. Chlamydia, intracellular or extracellular? Intracellular. Mycoplasma? Extracellular. Rickettsia? Intracellular, and Coxiella? Intra. So all of them are intracellular with the exception of Mycoplasma. Okay, good. You guys feel pretty good about that stuff? Okay. Well, in that case, we will drive on then. We'll spend I think probably all day today just finishing off this deck, and then tomorrow we'll start the nephrology deck, which I posted. I did remove 10 slides from that deck, by the way.
Haven't watched your class.
Okay, so we're talking about acid-fast bacteria today. Significance of it. First couple slides are a lot of detail about what the name means and the implications for this quality that they have. It's primarily due to their cell wall. So we'll revisit what the cell wall structure looks like for acid-fast bacteria like Mycobacterium and Nocardia, 2 genera. Mycobacterium is a large genera. It has lots of different species in it. Probably round about almost 200 species. Most of the species are environmental bacteria, lots of them, and many of them, like the NTMs, the non-tuberculosis mycobacterium, can cause opportunistic infections. That is a hard and horrible thing to study, but fortunately we're not talking about any NTMs. We're mostly focusing just on Mycobacterium tuberculosis, that one very famous species which is clinically relevant and important for us to have a good understanding of. So in this image, we're not looking at an individual cell, but we're seeing from a liquid culture of tuberculosis spigera something called cord factor, where these bacteria form these kind of serpentine cords or belts. This is sort of a virulence factor. It protects them from phagocytosis if they're able to do that. So it's usually used as an indication of cord factor. But if we did look at those cells and specifically looked at the cell walls, they would typically be gram-positive bacteria. So they do have a layer of peptidoglycan, but more characteristically, it doesn't really matter that they're gram-positive because we can't stain them. This cartoon a couple slides later shows that here's your layer of peptidoglycan on the outside, but you have this very characteristic deep layer of mycolic acids, 60 to 90 carbons long, that confers a lot of the properties that we're going to be talking about. So because of that, they are acid-fast. We'll talk about why that is specifically. But while we're on this slide, mycolic acids, between that and the peptidoglycan, you have this layer of arabinogalactans that are connecting those layers together. But this mycolic acid layer is what confers that waxy acid-fast property. They are aerobic. They require oxygen. They're very slow growing. You still have to culture them. Some of them can take weeks to culture them, so that is a major hurdle in the lab when we have to culture these organisms. For Nocardia, these are sort of similar to when we were talking about actinomyces last block. If you remember us talking about actinomyces, the one that was associated with these different abscesses and draining sinus tracts. The only difference is this is an acid-fast bacteria, whereas actinomyces will actually gram stain as positive. This won't though, because it does have mycolic acids on its cell wall. They're just not as thick. So we call it partial acid-fast, or weakly acid-fast. This is very slow-growing bacteria. It has a characteristic morphology. When you do an acid-fast or modified acid-fast stain on it, it has this sort of branching filamentous appearance that looks a little bit fungal. Also sometimes described as beading, just the way the stain is taken up by those bacterial cells. So filamentous, branching, you can see the branching morphology as those rods are dividing in chains together. Many, many different species. Not also opportunistic though, so we will only be focusing on just the genus as a whole, and not a whole lot of virulence for these. We don't get a ton of cases in the United States. Most of the cases that we do get are associated with opportunistic infections and kind of weird case reports. All right, drilling down a little bit more detail about acid-fast qualities and what that means. If you were to try and stain Mycobacterium tuberculosis or another mycobacterial species with a gram stain, the crystal violet stain that you initially apply during a gram stain wouldn't be able to permeate or permeabilize through that thick layer of mycolic acids. So for that reason, as soon as you go to decolorize the crystal violet, it'll just rinse all of it out and nothing will stain. For that reason, we rely on some different stains. We'll talk about these specialized stains, including fluorescent dyes like orabine and rhonamine, that can actually penetrate down through those mycolic acids and stain the cells. When you are decolorizing with one of those heavier stains like carbonyl fusion, you use a stronger acid solution to decolorize. And the reason why we call them acid-fast is because even that stronger acid is unable to lift that carbonyl fusion out of the cell wall. So it holds onto that stain and retains that pink, kind of purplish stain. And that's why we call it acid-fast, like hold fast with that stain. You can't decolorize it. We can apply different conditions to help force those bacteria to take up that stain. One of those is called a Zils-Nielsen stain, where you heat up the bacteria and that kind of loosens up that waxy coat a little bit and allows the stain to permeate. Or you can use a stronger acid, that's called a Killian stain, or a cold stain, force cells to die that way. But the reason why we go through all this is because we'll show you a testing algorithm, but we still cannot replace, in terms of diagnostics, we can't replace doing microscopy with PCR. There's a number of pitfalls with PCR. We can do them in conjunction together, but we can't replace it altogether. So we still have to do this. And it's also extremely rapid. You get an answer very quickly. If you find these bacteria in like a sputum smear from somebody you suspect has tuberculosis, it's extremely clinically relevant. You find these bacteria there, they're likely to have tuberculosis, and if they're symptomatic, So that would have immediate implications for putting infection control and putting them in isolation, which you would do for a TB patient. Real quick, this is a colony of Mycobacterium tuberculosis. Sometimes call those tough, rough, and buff for the color and for that texture. But they are kind of impermeable, so that waxy coating keeps them like a nice hard pellicle coating over those bacteria and forms. But this could take 2 weeks, 3 weeks to grow out this colony, so it does take a long time. With those stains, we've talked through most of this, but these traditional acid-fast stains, this heat-required one, Zil-Nilsson, and the cold stain came in. Both of them work by applying this carbofusion stain as a base, and if it is acid-fast, when you decolorize, it will retain that stain, it will retain that carbofusion stain. So you have a nice pink cell there. With a non-acid-fast bacteria, like any gram-positive or any gram-negative that we've talked about so far, that stain will lift right out and you'll get clear bacteria. Okay. Then you counter stain with something like methylene blue, this blue color, or green stain like malachite green, and everything else will take up that blue stain. So with these fields, you can see these acid-fast bacteria have this nice pink color, and everything else is blue because it's taken up that methylene blue stain. When you get a smear to analyze in the lab from a patient, like a sputum smear, it takes a very long time to go through that slide. We're supposed to analyze about 300 fields at oil immersion, 100x magnification. So that takes some time. And it's not super sensitive, you can easily miss these bacteria as well. If a patient has a heavy bacterial load, we'll see that, and again, that's useful. We have a scoring system, a semi-quantitative, where we'll score it as 1+, 2+, 3+, 4+, depending on how many bacteria we see. But we can culture up this organism and get culture positive and very easily miss these altogether on smears. So we have a sensitivity issue sometimes with these. For that reason, and trust me, so I used to spend hours like leaning over a scope trying to read these, and it is very frustrating when you don't see them, because it doesn't necessarily mean they're not there or the patient doesn't have a tuberculosis infection. So for that reason, fluorescent stains are super nice when you can do them, because it speeds up the process of identifying them immensely. You do these fluorescent stains which bind to those mycolic acids. This one, Auramine rhodamine stain, is nice. It actually has 2 different fluorescent colors. The Auramine is sort of like this greenish color, and the rhodamine is more of a yellowish, orangish color. But you can see from culture how easy it is to identify these bacteria. And then this is one that's done directly from sputum, so you can see a little bit of background fluorescence, but it's still super easy to see those organisms. In terms of lab identification, we will do PCR. We have some PCR tests for this. Some of those PCRs, like GeneXpert has a PCR cartridge for TB. Some of them will even detect antibiotic resistance genes, which is really nice too. But as I said before, you still gotta do everything else. So when we culture these, we will usually culture them on solid media, like this Lowenstein Jensen media. You can see those colonies forming, it's a slant, it's a different type, like a tube with this solid media on a slant. But those bacteria are growing up here. And then we also do a liquid broth as well. So these are midget tubes with a broth called, I forget what the broth is called, like a Middlebrook broth. But it has a fluorescent indicator in that broth. So while these bacteria start growing, if they're present in the patient specimen, they'll take up the oxygen and deplete the oxygen in these midget tubes. It's been diffused into that media. And then that will dequench a fluorescence marker if it's in that liquid media. So you'll get release of fluorescence, and then we have instruments like what's it called? Bactec 960 that'll detect the amount of fluorescence in that specimen as it increases and give you like an indication of whether or not you have tuberculosis or other microbacterial species. So that's a nice automated platform that we have. Length of time up to 6 weeks. Might be a little bit faster if you do a liquid broth, but it's going to take some time. but it is primarily how we do antibiotic susceptibility testing. So mostly not an option, not that you have to do this. For antibiotic susceptibility testing for Mycobacterium tuberculosis, it is super important. If you know anything about tuberculosis, you know that we have an issue with drug resistance. So we can do antibiotic susceptibility tests. Most labs will send that out to specialized labs to do that. But yeah, suffice it to say it has to be done, it's super important. And then lastly, we've already talked about microscopy, but super fast, very cheap, relative to PCR, and you can do those super sensitive fluorescent stains as well. This is an example of the algorithm, or a simplified algorithm for testing when you suspect a patient might have tuberculosis. So they would be presenting with signs and symptoms of tuberculosis, some respiratory infection. Maybe they have a travel history that indicates that they've been out of the country, something like that. You'd submit a sputum specimen down to the lab for TB testing. The lab would pre-treat it, which means they would do a digest and all the other cellular material, try to get rid of all the other bacteria, such as an outgrow mycobacterium. And then they would split that specimen up into different aliquots, different sections or parts. One of those would go into for culture, liquid and solid culture. Another one you would send to acid-fast staining, and then the other one you'd send for PCR. For the culture, fast forward a couple weeks, 4 to 6 weeks, you get your culture results, and you can send that off. You can identify definitively what that mycobacterial species is, and also send that off for drug susceptibility for ASTs. So it's a long process, yeah.
I just have a question, ma'am. I know we've talked about ASTs a lot, but I guess like let's say that we draw cultures. Are we supposed to order the ASTs, or do you guys automatically do them for like most common bacteria that are susceptible to certain antibiotics?
That's a good question. I don't know if you'd order an AST unless you have the organism that grows out of the specimen. But that doesn't mean you couldn't put in an initial order at the same time. It's just there's gonna be, as opposed to usually where you have like a 24-hour delay in overnight growth you need to do to incubate a specimen. You have lots of time with this. You have a month to sit there and wait for this organism to grow. And then if that patient is symptomatic, then you will want those results, but there's nothing you can really do to hurry up those results. You're going to prescribe empirically antibiotics and wait for those AST results much later. Does that make sense?
Yeah. Yeah, it does. I guess. Yeah.
Yeah. Okay. Let me know if you still have a question on that. We're going to talk about first line and second line treatments as well, and how that a little bit of the basics on what goes into that thought process. So in terms of pitfalls here, real quick, how you interpret these results as a clinician is important because you're evaluating the patient's clinical picture. If they are symptomatic and they get a positive PCR result, that's really good evidence. You know, that's a nice cohesive piece of evidence that they have TB. However, we have some interpretation issues, so these things can be interpreted with caution. If you have a patient who presents like they have tuberculosis and they're PCR negative, you would want to collect multiple specimens and resubmit again for testing, just to make sure that maybe their bacterial load is really low, maybe you've messed up specimen processing, something like that. And this is all while culture results are pending. And then if you have an asymptomatic patient, I don't know why you'd submit PCR in the first place. That's just asking for an interpretation nightmare. If you do that and you get a positive PCR test, the risk is that you over-interpret those results, or it could be due to any number of things. It could be contamination in the lab where you get a false positive PCR test. So yeah, it wouldn't really make sense to submit or request a PCR for an asymptomatic patient. You should have a good reason for submitting a PCR.
So in that situation, whether it's negative result but they're symptomatic or positive result but they're asymptomatic, you would get a new sample regardless of the situation. You wouldn't retest the same. sputum or?
Probably not, but that might vary, like the SOPs and the algorithms would probably vary from location to location. But we are just talking about sputum samples, so it's an easy to collect specimen type, it's not irrecoverable, it's not anything like that. If somebody does have symptoms like that, they would probably be submitting sputum samples frequently for monitoring drug therapies and progress like that as well.
And if someone is asymptomatic, what would be your reason for testing? If they're
That's a great question. I can't off the top of my head think of why you would wanna submit for a PCR test if somebody is asymptomatic for TB. Maybe travel history,
Proximity to other.
yeah, perhaps it's an exposure,
It would most likely be proximity and travel history, like a lot of the places that we will pick up going is actually organic,
yeah,
not to mention depending on your mission set, likely have close contact with populations
If-
they have it.
yeah, and there are some other tests that we could do if you do suspect somebody based on travel history and exposure. There are some other tests we could do as well. But PCR, like again, if you get a positive PCR test in somebody who is native, who has native sputum sample, they could have latent TB. And there would be other ways of diagnosing that. So, yeah, I wouldn't, PCR, it's just hard to interpret when you have a positive test result on something that's asymptomatic. Oh, and there's this little, I put it over the nucleic acid thing, it's not on your slide, but yet another little gene expert cartridge for simple MTB, Mycobacterium tuberculosis, XDR, so extremely drug-resistant tuberculosis. And this PCR is just, it's primers and probes are looking for a resistance gene for rifampicin, which is one of the first-line drugs for tuberculosis. It's a pretty good indicator that you have the resistant strain. Yeah.
How does this overlap with like the screening test like PPD or quantiferon gold?
Right. Yeah, we'll talk about that too, but with the tuberculin skin tests, the TSTs and IGRAs, interferon gamma release assays, IGRAs, those are looking at whether or not you have been infected with tuberculosis. Whereas this is, what's PCR testing? It's testing for the presence of that organism's DNA in the specimen, which would be more like an active infection. Okay, a little bit of, actually quite a bit of epi in the next couple of slides here, but in terms of the worldwide picture and the burden of tuberculosis globally, we have it pretty good in this country. That's sort of the takeaway for a lot of these slides. We try to keep our incidence rates low, a couple slides about that as well, but as you can see on this heat map, this is from 2021, some countries have a pretty high incidence rate of tuberculosis. Countries like South Africa, something like North Korea is lighting up pretty darkly, the Philippines, Indonesia, Bangladesh. I mean, there are some hotspots for tuberculosis, and for that reason, this is why we take into account travel history with tuberculosis. In terms of what constitutes an infected person, you can see this number, 1.7 billion people is probably a little bit higher for the number of infections. It's probably more like 2 billion. So the world population right now is 8 billion, so this is more like a quarter of that. But in your minds, if you think about that, a quarter of the world's population being infected with tuberculosis, does that seem a bit high to you? It does, right? It seems like shockingly high. But it's because we think that most of those infections are LTBIs, latent tuberculosis infections, where people have been exposed, but through this process that we're going to talk about, their body has contained it, and they are in a latent carrier status. So that's why this number is so high. Globally, 10.6 million active cases. So this is not LTBIs, not latent tuberculosis, these are active cases. We have about 8,000 in the US. If you do the math on that, we share a very low, disproportionately low burden of global active TB cases, and that's because of all the really rigorous screening and control programs we have in the United States. Similarly, if you look at the death statistics for tuberculosis, 1.6 million deaths globally, and in our country we have 500. So if we have about, 4% of the world's population, these numbers are just a small fraction of that. So we're doing pretty well in the US, that's the whole takeaway from that. I pulled some data I thought would be interesting for us to look at in terms of the United States. Sometimes data gives you a better picture of what's going on than just, talking about incidence rates. Here we're looking at 2021 data in the United States. Our goal for incidence rates is under 2.5 cases per 100,000 people. So if we look at the incidence rates of different states across the union, Alaska and Hawaii for 2021 data have really high incidence rates. Alaska in particular, very low population density, but they have a lot of cases. There are some reasons for that. For the lower 48, we have some states in 2021 that had a higher incidence. Texas at 3.4 is pretty high. Let's see, New York's got 3.4, high immigrant population there. Then if we move ahead to more recent data, I just pulled this is the most recent data we have for 2023. You can see there's been a little bit of shift. Now Alaska's even higher at 10.5 cases per 100,000. So that is a huge increase for that particular state. California's also had an increase. Texas has decreased a little bit, down to 4.1. And New York has increased now, so it's higher than Texas. So kind of interesting trends here. I like going on. CDC has a great website, great monitoring and surveillance program for TB. So we can learn a lot from just going on there. And then finally, this map is just plotting population density versus case count. So that gives you a truer picture of the burden of TB in these different states. And if you look at it that way, if you look at incidence and case count, states like Texas and California and New York have a pretty significant TB problem, but it's only relative to other states. Again, globally, all states are doing fine. Okay, so in terms of transmission, this is an airborne disease. When somebody has tuberculosis, like primary tuberculosis or pulmonary tuberculosis, they infect others by coughing, sneezing, laughing, talking, and those aerosolized droplet particles are inhaled by susceptible people, and they can establish infection that way. It's a bacteria called tubercle bacteria, very, very hardy. Important to know that not everyone infected with TB becomes sick, hence that high number of people worldwide who have TB. And it is possible to clear the infection right away too, so we'll talk about that more as well. It has a high association with AIDS patients, which is really, really unfortunate, because what do we know about HIV? What kind of cell types it targets? T cells, specifically CD4 cells. So as we talk about the pathology of tuberculosis, we'll see that those cells in particular are very important for keeping tuberculosis in check. So where there's high prevalence of AIDS, unfortunately there tends to be high prevalence of tuberculosis as well. Also crowded and homeless populations. If we look at these trends over time, tuberculosis cases have been dropping steadily, particularly since like the 1800s when we started having better hygiene practices, especially in the United States. If we look at from the 1950s going forward though, we see a huge drop in the 1950s. This is when we started to develop antibiotics like streptomycin in the 1940s and rifampicin and isoniazid in the 1950s. So when we had those drugs available, TB cases continued to drop in conjunction with a really robust infection control public health response to tuberculosis. So great success there. And then in both of these, they're basically showing, both of these charts are showing the same data around the early, around the 80s and 2000s. Actually, it's only the one on the left here. Look at the one on the left. We see a little bit of a spike here in the late 80s, early 90s in tuberculosis cases. What's going on there, do you think? Yeah, the AIDS epidemic, exactly. So we had defunded some of our tuberculosis control programs in the 70s because we considered it to be under control. And then the AIDS epidemic happened. So we saw an increase in in tuberculosis cases. Fortunately, we've been able to, with our treatments, the antiretroviral treatments for AIDS, we've been able to help control that problem a little bit better. Throughout history, I mentioned this was a big problem, particularly in the 1800s. This is one of the few diseases, just for historical interest, that was glamorized a little bit and considered to be an attractive disease to have, particularly in like Victorian era times. And particularly, there was like this kind of sexist association with women who had tuberculosis. Some of the signs and symptoms with tuberculosis were considered to be beautiful, like the anorexia, the rosy red cheeks, the twinkling, staring eyes, the glossy eyes. So yeah, that was sort of glamorized, and then even among men, if you look at some of these famous people who had tuberculosis, a couple US presidents, we have Chopin, we have Rousseau, Longfellow, Whitman, Simon Bolivar. It was sort of associated with creative genius as well, so, and like intelligence and, the best poets of the day had tuberculosis. So, for a while, that certainly hurt the ability for people, for infection control people to get help, convince people that you needed to get this under control. We eventually started seeing signs like this, though, just warning the population there was gross spit in public areas where there would be a high likelihood of infecting other people, so we saw placards like this on trolley cars, warnings about tuberculosis as a disease. And gradually over time, people became better about coughing etiquette and things like that. But for a while, in the United States, we had a huge tuberculosis problem. Alright, I'm talking about pathogenesis a little bit now. We talked about how you get it. You inhale these Mycobacterium aerosolized respiratory particles. They get deep into your lungs, into the alveoli, the air sacs in your lungs, and they are engulfed by alveolar macrophages. It is possible to clear the infection right away, depending on the infectious dose, how much bacteria you inhale in. But most of the time, if you inhale a sizable or appreciable infectious dose, these bacteria will replicate because they're intracellular bacteria and they survive ways of escaping death in macrophages. So they'll replicate, and then they can, if they're not cleared within a month, you can start getting some involvement with the adaptive immunity, and that contributes to the pathology of this disease as well. So, within that month-long window, 2 to 4 weeks, when you start getting the T cell response, the cell-mediated response, at that point you develop this very unusual containment strategy that occurs when your body really doesn't know how to deal with a disease. This is a granuloma formation. So we've been infected with tuberculosis for thousands of years probably. There's historical evidence that we've had cases back to like 600 BC. So our body has been confronting tuberculosis for a long time, and we know that if you don't contain this bacteria, if you aren't able to clear it with your innate immune system, that you run the risk of dissemination, and that has a very poor prognosis. So what our immune system has evolved to do when it can't clear an infection with innate immunity is to form these granulomas around the bacteria, to cordon them off and prevent them from being disseminated across the body. When you do that, when you successfully form a granuloma in the lung around tuberculosis bacilli, that is when you are considered to have latent TB. And for all intents and purposes, it's not a terrible condition to be in. You're not infectious, you're not symptomatic. You might have lost a little bit of lung tissue with that granuloma, but the job is done. You've successfully cordoned off those bacteria. However, the problem there is, we'll talk about this more when we look at some of these images, but within those granulomas you can have surviving bacteria. It doesn't necessarily kill the bacteria. So when or if those granulomas ever lose their integrity, stop being able to maintain those granulomas, and those viable bacteria get out of the bacteria, or out of the granuloma, they can be taken up into the bloodstream for disseminated TB, or they can cause another, like secondary case of tuberculosis if they infect the lungs again. So those are some issues that we have to be concerned about. In terms of virulence, no toxin production, it's mostly just the body trying to contain the infection that causes a lot of the symptoms that we'll be talking about. But the ability to inhibit phagolysosomal fusion is considered a virulence factor, otherwise they wouldn't be able to survive in macrophages. And this ability to persist in macrophages for decades with latent TB is unique to tuberculosis. Not a lot of things can do that. And then reactivate down the road when you are most vulnerable, most susceptible. Let's just finish off this one slide and then we will, actually, let's do this. Let's take a break now. I've got 8:47 and then we'll come back and start talking about the immune response a little bit more detail. 8:57.
I remember him.
Well, yeah, or like, you know, one back in the day, you know, we had
I had this, my mail. You run over, I have not even last 4 years.
Those things are getting.
I rescue that.
They made us one.
I'll bring you over some phone. So I have two options. And I'm doing the gotcha this time. I know you can do the gotcha gun, but I also have bread. This is my first time doing gotcha gun and not eating red pepper trees. So it's whatever, you know. I'll bring the gotcha gun and do but it's only $4. Unless you want it super spicy. Even a quarter cup is not. I don't think it's not. And I think it should be disappointed on a dog. Yeah. Disappointed.
I don't think ease of something is disappointed. Unless it's like, why am I doing this long?
They still have like they put it
Yeah. Now I just gotta get caught up on here. Like study. You keep me on. Yeah. Like it's just like road utilization. You know what I mean?
That was a lot. Yeah, it is this rough memorization, but it takes a lot.
I think it's just like one of those you need to spend more than 2 days a week looking at it. Okay, and then you start to kind of fall into place.
Nothing, like, you can stop with that thing, like,
Made into it.
but they did. It was a
You would cost just some time for the semester.
Yeah, exactly. That's kind of what I'm at. It's like, if I just burn it,
so they had some of the later to do. Or even somebody
I'm pretty sure it's just a cold. One day I was just dragging it for 4 hours put away. I'm curious if everything's going around. Yesterday it started like a little bit.
Yeah, that weekend.
Something
Try to take an emergency.
Yeah.
I think it's good now. It really does. And then that's all I do is like, around when I wake up, it's just one that I do every day. When I feel like, or I feel like someone around me is and if it doesn't mind. Okay. I just little tablets. This provide with generic training.
You should have had something on there. That was probably the best.
It was, yeah. There's a ton of people though, I mean, yeah, so many people that teach We've got 8:58. So, get going again. Talk about the immune response in a little bit more detail. Aren't you guys glad you had immunology in class 1? Now we can understand this a little bit better, what happens here. Alright, so as I mentioned, you inhale these bacteria, they get into your lungs, you might clear them with your innate immune system, depending on the infectious dose and your own immune status. But if you don't, then the adaptive immune response is going to kick in. What are the two arms of the adaptive immune system, again? The two arms. So one of them, yeah, so one of them is cell mediated, and that's they're T cells, yes. So T cells with the cell mediated response, and then the other arm was what? Humoral. So we're talking about antibodies produced by B cells, right? With antibodies, those are really good at clearing extracellular bacteria, extracellular pathogens, toxins, things like that. But when you have intracellular bacteria, things like mycobacterium, things like the rickettsias, coxiellas, listerias, some of the things we've been talking about, then the cell-mediated response is going to be super important. So that's not to say you don't raise antibodies to Mycobacterium tuberculosis. You do. You raise them against some of those antigens, surface antigens on those bacteria. But it doesn't do a whole lot to help clear the infection. It's a cell-mediated response, CMI, that does the heavy lifting. And that's because of CD8 T cells doing their things, what those do to clear infections. Yeah, cytotoxic T cells that kill infected cells. So these infected cells, if everything's working properly, you get a presentation of those antigens on the surface, and then MHC class 1 or 2? 1 will be recognized by those CD8 T cells, and then those CD8 T cells will destroy those cells. So that's important for clearing TB infections. And also CD4 T cells that do a lot to coordinate that response and form these granulomas that we'll talk about it as well. Super important. This is implications for vaccine development too. Fortunately, we can't give a nice, super clean vaccine for tuberculosis, or we would have done so. There is a vaccine for it, but it's a live bacteria vaccine. It's using Mycobacterium bovis, a related species, to do that. And the reason why we have to do that is because we need to stimulate the cell-mediated immune response. That's why it's so important. So we'll talk about that later too, but I just want to put that out there. Okay, so the adaptive immune response, when and if it kicks in for tuberculosis about a month down the road, it will do a lot of good work in clearing this infection. It can clear it, but as you proceed into, if the bacteria start replicating and you get an actual infection going in the lungs, there could be some significant pathology as well. Those immune cells will be releasing a lot of cytokines, you will get inflammatory processes, And then the granuloma formation as well is damaging to the lungs. Wherever those granulomas form, it's basically destroyed tissue, you're never gonna get it back again. So you can easily afford to have a granuloma or 2 or 3, but when your lungs start getting riddled with granulomas, then you have decreased oxygen carrying capacity in your lungs and a whole bunch of other things. So double-edged sword, that's the whole point of this slide. Immune response is important, but it's also damaging tuberculosis. This slide is the one that we're going to use to kind of drive and steer our way through infections because it gives a percentage, kind of maps out the likelihood of proceeding in different ways, all the ways this very complicated infection can go. But when we're talking about primary infection, we're talking about a relatively small percentage of cases that will actually proceed to real infection, primary tuberculosis. Majority of those will be eliminated. 70% of people are just going to be able to control that infection before you get symptomatic tuberculosis. For those who do get an infection where these bacteria are starting to replicate in the lungs and draw attention from the immune system, you can start getting some damage going. This is when you will start getting symptoms of tuberculosis. So you'll have some coughing, fever, perhaps, malaise, feeling poorly, and things like that. And at that point, this is when you start getting recruitment of these immune cells into the area to form granulomas. Looking a little bit more carefully at what these granulomas are, it's just a pylon of different immune cells that happens in the body. So very organized. We've got lymphocytes, macrophages, giant cells, Langerhans cells, pylon, fibroblasts, capillaries, a whole bunch of cells with pylon. Very coordinated, very organized. And they start building up around these bacilli. In the center of these granulomas, it should be a pretty toxic environment. Over time, it starts getting liquefied. Anything that was viable in there gets destroyed, with the exception of some of these hardy resistant bacteria. They may or may not die in those granulomas. So when you have latent TB, that's when you have viable bacteria that remain in those granulomas. Lucia had mentioned this analogy of a ticking time bomb. So that's sort of what LTBIs are. You're walking around with a controlled infection, but viable bacteria in those granulomas. As long as your immune system is able to maintain these granulomas, which do take constant maintenance, this is not like something you can just walk away from. You have to constantly maintain them, CD4 T cells in particular. As long as you're able to maintain those granulomas, you should at least be able to contain any viable bacteria from escaping and going into the bloodstream. But when you get old, what happens to your immune system? So even if you live to be super old, eventually your CMI is going to give out and you're going to get tuberculosis. Yeah.
So it's most of the CD4Ts that are responsible for.
Yeah, it's primarily coordinated by CD4 T cells, I think. But there are other cells involved as well. So a lot of cytokine signaling and communication between these immune cells and epithelial cells. My older brother, this is what he did his dissertation on. He wrote his dissertation on granuloma formation and T cell response to TB. So he used to call and talk about all of his experiments and all of his frustrations with his experiments, and it was incredibly boring to me then. But now I think it's pretty interesting. And if you want to do a long course for a PhD, study tuberculosis. It took him 8 years to finish that because of how frustrating it was to work on his experiments. He was using mice as a model organism for TB. But yeah, he could tell you all about that. So yeah, your CMI keeps these things maintained over time, and over time, we're talking about decades. You contract this, let's say, when you're 20 years old. Just can imagine over the decades you having these granulomas in your lungs. I mentioned aging, but what else do you think might cause your CD4 T cells to start decreasing in their ability to maintain granulomas? Immunosuppressants, yeah, immunosuppressant therapies or drugs. Anything else? Yeah, aging. So again, when you lose your immune your CD4 counts start dropping over time, this invariably is going to start being impacted, your ability to maintain those granulomas. Very, very important thing to remember. At that point, when your adaptive immune response kicks in, when you have your cell-mediated response and B cells involved as well, this is when you start having positive tuberculin skin tests, the TSTs, and those interferon gamma release assay tests, IGRAs. Those will be positive at this time. The IGRAs we'll talk about too, but this is basically an assay that's taking your T cells from blood specimen and stimulating them with an antigen from Mycobacterium. If they release cytokines, they've already been pre-exposed, so it's an indicator of exposure to tuberculosis.
It might be a little bit irrelevant, I'm just curious. After those bacteria get, surrounded by those granuloma, for decades, how do they stay alive? There's nothing inside for them to survive.
That's a great question, and they're just very hardy bacteria. They've evolved to do that. This particular species, Mycobacterium tuberculosis, not the other mycobacterial species, but this species has evolved to slow down its metabolism. It doesn't divide very much, it doesn't transcribe a lot of genes, it's not transcription active. So they kind of go into this quiescent, dormant state in the granulomas, and that's how they can survive for all that time period. Yeah, John, sorry.
Is there a time where we know aging is gonna cause this probably relapse and come back, where we just go in and manually try to remove it, so it's like net gain positive to get rid of it?
You don't normally do that. Somebody else had asked this question too, like, first of all, a lot of people who have granulomas and LTBIs are never diagnosed, or they don't have chest x-rays indicating that they have those granulomas, which oftentimes are hard to see on chest x-rays. They're easier to see if they get calcified, and they're easier to pop up on chest x-rays. But they're not a problem, so you wouldn't go in and surgically excise them unless you had to. You're not gonna be able to treat it with drugs though. So I have read about surgical excision when you start having like decreased lung capacity when it's that bad, but otherwise don't surgically remove those. Yeah. And you had a question too?
Yeah, I was gonna ask, and I think you answered. I was gonna say, even if you gave antibiotics, would it even kill it? Since is it also protecting it from antibiotics? I guess in a way.
That's a really good question. And it's a little out of my depth, but yeah, the granulomas around them does not help at all for antibiotic penetration. But when they do have latent TB infections, they might be put on like a lifelong course of isoniazid. I'm not entirely sure about that, but or one of the other antimicrobacterial drugs. But you do have a lot of thought and consideration that you give to the penetrating power of whatever antibiotic you're prescribing for getting through the granuloma, through the cell wall itself with all those mycolic acids, and actually doing its thing, targeting bacteria. It would be daunting. Okay, so you formed your granuloma. This is sort of narrative style. We're past pulmonary tuberculosis, you've survived that, and you have granulomas in your lungs now. This is a little bit reiterative, this slide, but a couple things to note here. These percentages, you have 5% of healthy people who will have active disease within 2 years, and another 5% at some point in your life. Those are pretty low percentages, so that's a testament to the fact that this immune strategy that sounds kind of damaging and maybe not great is actually a pretty good strategy if such a low percentage of people undergo reactivation. Hopefully you will just die of natural causes and you will not die from reactivation. You'll die with those intact granulomas. That's sort of the body's strategy, right? We're not going to live forever. You just need it to last until you die of something else. So that's the strategy there. I had mentioned too, when you are in this latent form of tuberculosis, it is kind of okay, notwithstanding that you have viable bacilli in these granulomas, but you are not shedding. So you could be sitting here in this classroom and not be a transmission risk to the person next to you. And you're not symptomatic. You're simply positive on those tests. So that would be an indicator that the PPD test would be positive and IGRAs would be positive. And you might see that on a chest x-ray, but you will not be symptomatic. And hopefully, like I said, you lead a perfectly healthy life with your granuloma riddled lungs. However, if you don't, we talked about the maintenance of these structures and what happens if they give away. If something happens, particularly if these bacilli are released near a blood vessel where they can be taken up and disseminated very rapidly across the body, this can cause a lot of problems and it can cause a very different course of disease. So first of all, this is we have a couple pictures of tissues and organs with these granulomas showing the caseous center, this cheese-like center that forms there. This is what that looks like. If this granuloma ruptures, these bacteria can be released and disseminated. And you can get this reactivated, disseminated form if it's taken up in a bloodstream, where virtually any organ system you can imagine in the body can be affected by these bacteria. Wherever they go, if they lodge in your heart or your brain or your lymphatics or your bone, the immune system is going to have the same response and you're going to have the same immunopathology. So your body is going to try and form granulomas wherever those bacteria are, and that causes a lot of problems and a very poor prognosis as well. Rapidly fatal if undiagnosed and untreated. Here we can see a pie chart of different body sites that are involved with tuberculosis. As expected, most of these would be pulmonary cases. Even secondary reactivation cases sometimes don't make it outside the lungs. They can stay in the lungs. But if they do disseminate hematogenously, these are some of the systems that are more frequently affected. The lymphatic system is fairly commonly affected. The pleuritic tissues, what's encased in the lungs, are infected sometimes. Bone and joint infections, you wouldn't normally associate with tuberculosis, right? But this is a picture of somebody who has an infected ankle bone with tuberculosis. So there's a granulomatous process, inflammatory process going on here. Yeah, and you can get granulomas just kind of riddling the body. This slide, there's not a lot that we haven't covered, so I'll skip over this, except for this bullet right here, that when you have disseminated TB, also known as miliary TB, this is a very small percentage of TB cases. So generally speaking, if you're immunocompetent, you are not susceptible to disseminated tuberculosis. You get a handle on it fairly quickly. It's people who are immunocompromised that have a harder time containing this infection. This is just kind of interesting because it's a mummy that had a bone infection called pot disease, tuberculosis bone infection. We still see this presentation today, but it affects the vertebra in the spine, causes that kind of like disfiguring disease. A couple more pictures of what these granulomas look like. Here you have some disseminated tuberculosis in this woman's neck. Here you have some sinus involvement. You can see cervical sinus involvement. A chest X-ray, primary tuberculosis. These look like airways, probably from an infant who may have died of tuberculosis. They're very small, but the circles, those yellow and red circles, are around what are called Gone complexes, which are basically calcified granulomas. You can see how large they've gotten. This histopathology shows you the organization and the architecture of all the cells that are involved in making these granulomas. And then here we see kidneys and other organs that are just riddled with small little granulomas that start impacting organ function with disseminated tuberculosis or with pulmonary tuberculosis as well. Alright. Lastly, talk about secondary or post-primary tuberculosis. We usually think about, when we think about secondary tuberculosis, we think about reactivation when those granulomas have given way and viable bacteria start replicating actively in the lungs again. That is possible for secondary tuberculosis, but you can also get re-exposed to another person and they can transmit the bacteria to you for reinfection. So that too will cause a similar pathology. You're not immune or resistant to getting a new infection. When your body sees these bacteria that they haven't seen in, let's say, like 10 or 20 or 30 years, there will be memory T cells and memory immune cells that recognize these bacteria. And because of that, you'll have a fundamentally different immune response with reactivation TB. That immune response is usually worse, and the pathology is usually more severe because of that, faster and more severe. For one thing, the granulomas will form faster, and you can get these distinctive cavitations in the lungs that you don't really see with primary tuberculosis. The cavitations are basically hollowed out parts of the lungs that you can easily see on chest x-rays, and that would be diagnostically useful for secondary or post-primary tuberculosis. So cavitations, some more severe symptoms in the lungs, pleural effusions, where you get a lot of liquid and fluid in the lungs building up, empyema, and these severe symptoms that are associated with active reactivated tuberculosis, night sweats and chills, fatigue, anorexia, and hemoptysis. What is hemoptysis? Coughing up blood. Yeah, exactly. So these are usually the more severe disease presentations. Host has been sensitized to those antigens already. We've revisited, or we are revisiting some of these diagnostic tests now. Culture is not on here, but we would be doing cultures as well. When people have this presentation, again, that initial AFB smear on sputum samples is going to be really, really important initially, as well as those chest x-rays. They may or may not be positive, but if you see things like cavitations or granulomas on chest x-rays, and they have symptoms, that would be concerning. Positive skin tests, we'll talk about that in just a second. These IGRAs, which, again, is just telling you whether or not your T cells have seen this antigen, these antigens before, and there's expressing cytokines, particular interferon gamma specifically. And then the clinical picture is really important as well. So all of these things together is sort of the entire clinical picture that's used to evaluate somebody, whether or not they have tuberculosis and whether or not it's active or latent tuberculosis. This is a handy little algorithm for you guys to take a look at. I don't think we need to go over it in a huge amount of detail, but the different disease states on the top that are represented on this are sort of helpful to guide you in your understanding of these different aspects of the clinical picture. We have an instance where a person has been exposed to tuberculosis, but they've cleared the infection. In those persons, for example, they would not have a positive PPD skin test, they wouldn't have a positive IGRA, and certainly they wouldn't have any of these other indications of TB disease. So their innate immune system is cleared without the adaptive immune system getting involved. If you did have a minor infection but cleared it, you successfully cleared all the bacteria in your lungs are free and clear of this organism, then you would be positive for those skin tests and positive for the IGRA, and everything else would be negative. Latent TB infections, a little bit, well, pretty much the same. You're not infectious, you have a positive skin test and IGRA. But the question that you were asking, Mary, about whether or not if you were latent, you would be on antibiotics. You might be on preventive therapy, but I don't know exactly what that entails. And then if you do get subclinical TB disease where you have granulomas still, but you have actively dividing bacteria that have gotten outside of those granulomas, in that case, you might get a positive culture, you might get a positive sputum smear, but this is where it becomes most challenging to diagnose because you may or may not be able to find those bacteria. Subclinical, so not really symptomatic, but you have some bacteria in the lungs. Difficult to diagnose. And if you could diagnose that, multi-drug therapy at that point, which we'll talk about in just a second. And then active TB disease, everything's positive, and you're on multi-drug therapy as well. Okay, we've mentioned this tuberculin skin test before. Most of us have probably had it in this country. We rely on this test as a screening tool heavily. But what this is checking is whether or not you have an immune response to this antigen. It's a purified tuberculin antigen when it's injected intradermally into your skin. So it's a delayed, type 4 delayed hypersensitivity reaction that if you've been exposed before, you start mounting over the course of 48 to 72 hours what's called an induration, which is a raised, elevated, hardened spot in the skin where you have a bunch of immune cells coming into that area. And how we measure this indurated spot on skin is simply by taking out a ruler and literally measuring it for the size. And we take into account exposure risks, like whether or not you've had contact with known infectious individuals, whether or not you have chest x-rays that are positive, maybe travel history, I don't know. Oh, that's with the next category. Or if you're dealing with somebody who is on an immunosuppressant drug or something like that. Those are considered to be pretty high risk factors. So when you interpret the size of that induration, all you would need to have is an induration over 5 millimeters, and that would be a positive interpretation for that skin test. With the next category down, these are lower risk things, simply working in a high prevalence country or travel to a high prevalence country like South Africa or Russia or some other place. Prison populations where they have higher rate of TB, nursing homes, healthcare workers, so 10 millimeters or more. And then finally, if it's just any one of us, let's say we don't have a travel history, any normal one of us, we would take a pretty large induration in order for that to be interpreted as a positive TST test. So that's how those are interpreted. Alright, and then the vaccine. Did you have a question, Justin?
If you're already vaccinated,
Something will happen, unfortunately, yeah, and that's absolutely key, yeah. So let's talk about what happens if you are vaccinated and why we don't vaccinate in the US. There are probably some people in this room who have received this vaccine before, the BCG vaccine. If you were born outside the US, chances are good that you've received it. A lot of countries choose to vaccinate their population as a way of controlling against tuberculosis. But here, we don't. Why do you think that might be? Somebody raise your hand. Yeah, Pat.
We don't have a high risk of getting TB.
Exactly, yeah, so it's a little bit of a calculated risk analysis. You had something to add to that?
I would say the skin test would always be positive.
Exactly, so those are actually the two major reasons why we don't vaccinate in the US. One is we're low risk, we have that incidence rate on average of like 2.5, low incidence rate. And two, we lose the screening capability of doing a skin test because it's going to be positive, more than likely. You'll get a positive skin test if you've been vaccinated with this live bacteria vaccine, the seal pill that you're on, which is again is Mycobacterium bovis that you're injecting into somebody. So you get that nice T cell response, that's how you get protection, but you're going to probably be positive on your TSTs from that point on. And that's why we don't vaccinate in this country. There are some instances in the US where we will vaccinate with BCG. This is a little bit of an FYI slide. People who are at higher risk, healthcare workers, might be offered the vaccine, for example. Lab workers who are doing research with tuberculosis. But since this is a live bacterial vaccine, we do not want to give it to immunosuppressed people, do not want to give it to HIV positive people. This can be fatal in those people because those bacteria, even though they're relatively avirulent, can replicate if you don't have a competent immune system, so you can actually kill someone vaccinating them that way. Pregnancy, usually not a super good idea if you can avoid it. Better to avoid it. And then in terms of first-line drugs, if you have a positive case, this acronym is sort of what is helpful to memorize. It's RIPE. So rifampin, isoniazid, pyrazinamide, and ethambutol. These are the primary first-line drugs against tuberculosis. You should probably know them. You'll definitely be tested on them next semester in pharmacology, Part 1. But with all of these, we have different mechanisms of action. We have generally some pretty appreciable side effects. That's why I put this column here. You don't have to memorize these side effects, but they're not the nicest drugs in the world. Some of these have been around for a long time. They all have penetrating power in order to reach these intracellular bacteria. And all of them have been abused, at least early on, if used as monotherapies in particular. So getting to this slide about antibiotic resistance with TB, part of the reason why we never prescribe monotherapies for TB cases is that you do get resistance pretty readily with TB isolates. So we have poor use of monotherapies, that's one contributing factor. Poor adherence or poor compliance, what do you think that means? Yeah, if you were prescribed any one of those first line drugs in the first place, you're gonna be on those therapies for very long, like minimum 6 months, probably much longer.
I was gonna say, I was on it for 4 months.
Oh, wow, okay. Do you remember which drug?
It was.
Or drugs, I should say. Okay, yeah. Yeah, so poor patient compliance, if you're on anything long-term, patient compliance drops off a bit. In the US, we have something called DOTS, directly observed therapy. Have you ever heard of that before? Oh, wow, okay, so we take this.
That I give you your meds and I want you to take it.
Yes, like that's how serious we are about being compliant, because the risk is if you're putting somebody on a drug combo therapy and they have to be on it long-term, low compliance, but very high risk, what you're gonna do is select for resistant strains, and then if they become active carriers of tuberculosis, infectious, then you're gonna be spreading drug-resistant tuberculosis strains. We do not want this in this country. We're super serious about that. So we do something called directly observed therapy, where we watch our TB positive patients take their drugs to make sure that they take those drugs. Pretty hardcore compliance.
How are we doing that for 6 months?
I do not know.
FaceTime.
Yeah, I would be curious to talk to a healthcare provider who's had to monitor that, but I know that is a thing with tuberculosis. Yeah, I don't know. You kind of do, going back, I'm sorry, my mind just wandered for a second, but going back to that early slide where we looked at the fact that a quarter of the world's population has latent TB, and only 5 to 10% of those cases will ever reactivate, right? What's 5 to 10% of 2 billion? That's between 10 to 20 million people walking around with active TB worldwide. That's like, the entire population of LA or the entire population of New York walking around with active TB. We simply cannot afford to have that many patients walking around globally with the risk of picking up antibiotic resistance. Some of these strains are extremely drug resistant. There's hardly anything we can use to treat them. We have identified PDR or TBR TB strains. Those are pan-drug resistant and total drug resistant TB isolates, virtually untreatable. There's basically nothing you can do to treat those patients. So that's why we take this so seriously. Even XDR TB is still extremely rare, but when we see XDR TB, this is when you're resistant to the mainstays of the first line, so isoniazid and rifampicin, and one of these fluoroquinolones, and then one of these injectable second-line drugs. With these second-line drugs, like these are some aminoglycosides, I think. Those are not good drugs either. They have nasty side effects. So to have those start failing is really problematic as well. All of these are problematic, but it's just, it's a big issue. And then with MDR, this is the lowest class, just resistance to either isoniazid or rifampicin. Second line drugs, this is purely FYI, just good to know. This is like the regimen that is developed for cases is a tailored bespoke regimen that's developed by a team of experts usually. So TB docs will weigh in on this, other professionals, control groups, and they will decide which antibiotics to administer and how long, and then that patient will be monitored to see whether or not they're able to clear that infection. Okay, so that's it for tuberculosis. Just a couple more slides today. Talk a little bit about MAC organisms. these are not tuberculosis organisms, but it's a different complex of organisms that are associated with opportunistic infections, particularly with AIDS patients. That's how they were initially identified. As I mentioned earlier on, most of the 190 plus species in this genus are environmental bacteria. So these organisms, Avium, Intracellular, and Chimera, are environmental pathogens that we tend to pick up in opportunistic settings like healthcare settings. Fortunately, transmission is from whatever the environmental source is, so inhalation, inoculation, wounds, things like that, ingestion. We don't transmit person to person because these bacteria do not establish deep respiratory infections, so they don't get into our aerosols. Particularly at the height of the AIDS epidemic, before we started to get that under control, these MAC organisms were a bigger problem. Now we've reduced the number of cases that we see, much lowered incidence, thanks in part due to heart therapy. And now other causes of death have replaced it for AIDS patients. Some of these are intrinsically resistant to some of those anti-TB drugs. But otherwise, the main thing to know about the MAC complex organisms is that they're associated with immunocompromised patients, particularly HIV patients, and they're opportunistic. We also have one more species in this genus that we want to talk about, Mycobacterium leprae. Anybody ever heard of this guy? Yeah. Oh, you guys know about the Armadillo Association. Okay, we're pretty good at reservoir species. Yeah, so the reservoir, well, first of all, what disease does this cause? Leprosy. Anybody ever been stationed out in Hawaii or lived in Hawaii? A couple of you guys? Ever been to Molokai? Yeah? Been to Molokai? Anybody know about the historical association between Molokai and leprosy?
Leper colony?
Yeah, major leper colony. It's a very sad part of Molokai's history, that they used to ship leprosy patients off to this little island in the middle of the Hawaiian island atoll and basically make them stay there for the rest of their lives because it was such a stigmatized disease. And they considered it to be catching, which is ironic because it is actually a really difficult to transmit disease. You know, if you've been down to my office on the first floor there, we have a nickname for ourselves. We call ourselves the leper colony, which is not a nice thing to say, but we are separated from the rest of the IPAC faculty. So, just, yeah, inappropriate, super inappropriate. But we are cordoned off. In terms of incidence these days, this is really a pretty rare disease these days. We used to have, even 30 years ago or so, 5 million cases. This is global. Now we have about 250,000 cases, 2024 data. There are some hot spots, India, Brazil, Indonesia, where with higher incidence rates, but in the US, 227 cases a year on average, give or take. So very uncommon here, but it is good to know about. There's a wide known, I guess, association with this reservoir species here in the US that is endemic to this part of the country. So it's like eastern Texas and western Louisiana. We have the 9-banded armadillo up there. You can count the 9 bands on this armadillo. If you go up to Guadalupe Park, you see these things all over the place. I actually ran over one accidentally in my car. I know, I felt really bad about that. But yeah, we have a lot of I felt terrible. We have a lot of these in Texas. The carriage rate though, if you're going around playing with armadillos, is a bad idea, but the carriage rate, just FYI, is not that high, even in one armadillo species. It's like something like 1 in 7 armadillos carry leprosy, but you can contract it that way by interacting with these animals in a way that you shouldn't. And if you do contract the disease itself, you can transmit it person to person, but it is an extremely inefficient way of transmission. It's usually by bodily fluids, so close, prolonged contact takes a while to transmit. And on top of that, most people are naturally refractile to disease. 95% of people would have a hard time getting it in the first place. So just because our immune system is pretty well adapted to clear clear these bacteria. So, when you do manage to establish an infection though, because of how slow growing Mycobacterium leprae is as an organism, it can take a very long time to progress to having symptoms, like 20 years. This is one of the longest incubation periods known. Could be shorter than that too, but it does take a long time. Generation time for these bacteria is extremely long. In terms of presentation of leprosy, which is also known as Hansen's disease, this is a disease that primarily affects the skin or the nerves, nerve endings, causes nerve degradation, it's a hallmark. But there are two different forms of this disease. The first form is considered to be the milder of the two, and it is directly related to your immune system's competence at carrying these bacteria. If you have a robust cell-mediated immune response, you're able to keep the bacterial load down, and you'll have some granuloma formation, but these symptoms are milder than this other form, which is called the lepromatous form. Low bacterial load, paucibacillary, same thing. Pauci meaning few or scant, and then bacillary. So few bacteria is associated with tuberculoid presentation. With the tuberculoid presentation, over time, people will develop some hypopigmented skin macules. They may or may not, but that's a common presentation. On the cooler parts of the body, that's because this bacteria likes cooler temperatures. It grows best about 5 degrees cooler than our body temperature, which is the body temperature of armadillos, by the way, around 32 degrees Celsius. Peripheral nerve involvement leads to sensory loss, so sometimes patients will get like a tingling sensation in their hands and other extremities. With the lepromatous form, you have a higher bacterial load, a worse CMI response, a more sluggish CMI response. And because of that, you get some of these more severe symptoms. Widespread disfiguring skin lesions, these nodules, plaques, epidermis. This gentleman on the middle picture here has the lepromatous form of tuberculosis. You can see his nose is a little bit disfigured, so there's involvement with the soft tissue structures in his nose. And also he's missing his eyebrows, which is a unique characteristic of the lepromatous form. Also has a tendency to cause leonine features in the face, cat-like features. And then this gentleman also has the lepromatous form. You can see these kind of like cauliflower type swellings on his ears starting to form. Again, cooler spots of the body, you get these deformations and disfigurations. And then also in the hands, sometimes I don't have a picture of that, but you can see individuals where it affects their hands, they get gnarled hands, sometimes they lose those digits altogether. Very difficult to diagnose, and that's because your exposure could be like decades earlier, so you might not have any indication that you have it. But if you're in one of those hot spots, those 3 countries that are more endemic, perhaps they're better at empiric diagnosis. It's also difficult to find this organism on a smear, particularly if it's this paucibacillary form. So I don't know if there's a PCR for it, but with skin smears, there's a chance of somebody being positive for it and not being able to find it on a smear, even if you do like a fluorescent smear. So a little bit difficult to diagnose. This would probably be very important, looking for a combination of skin lesions and nerve function. But you would have probably more bacteria with this multifaceted form, as you expect. There's a hidden slide. If you have it in your deck, great. If not, don't worry about it. But it has 2 clinical vignettes in that slide. Does anybody know what slide I'm talking about? Okay, totally FYI if you want to read through those, but it gives you a better, more in-depth picture of like a classic lepromatous presentation versus a tuberculoid presentation of leprosy. And we will finish up with the last organism we're talking about in this deck, Cardia. This genus has a bunch of different species, as I mentioned, so we're not going to talk about any of them in great detail, but these 2, Asteroides and Resilience, are some of the more clinically relevant species in this genus. In terms of transmission, this is an environmental bacteria. It's not a commensal, it's not endogenous flora, so you acquire it through inhalation, ingestion, or inoculation. And the disease process that it forms depends on which one of these routes of entry you get. not considered to be infectious or transmissible person to person. Going back to the morphology of this bacteria, partial or weakly acid fast, again, takes up stain differently even than Mycobacterium, but it does have that beaded appearance, long chains, branching morphology, all characteristic for Nocardia. Slow growing like Mycobacterium. When you grow it up on a culture, you get these kind of rough, chalky looking cones. And if you look closely at some of them, they look very much like growing up an old form of fungi. So aerial hyphae extending upwards out of the plate. It's characteristic for Nocardia. But they are still bacteria. It's important to know they're not fungi, they're still bacteria. And opportunistic infections, if you inhale it, you get this bronchopulmonary disease presentation, cough. Can be fairly severe, depends on the inoculum, and it depends on your immune status as well. Oftentimes you can get exposed to Nocardia and clear it. So if somebody is diagnosed with Nocardiosis, you would want to make sure, like run a full battery on their immune system, make sure they have a functional immune system, because most people can clear these infections without incidents. So pulmonary forms, cutaneous forms, you get inoculation through breaks in the skin. With these cutaneous forms, I've got a couple pictures there. You can see a mycetoma, which is an infection, like a deeper tissue infection. This is the same term that we would apply for some deep fungal infections too, just FYI, but Nocardiosis, cutaneous Nocardiosis is associated with these mycetomas as well. Cellulitis that we've talked about before with staph, and subcutaneous abscesses. This is really, really big. Abscess association and Nocardia. They have this ability to form abscesses, which is particularly serious when you have CNS involvement, and the bacteria can make their way up into the brain and cause brain abscesses. One of the more fatal outcomes for Nocardiosis, and one that you want to pay attention to. Symptoms are not going to be super helpful. These are the most generic symptoms you can come up with, but again, in general, it just depends on the route of exposure. This child had no underlying conditions, was a perfectly healthy child. But just in terms of like an interesting case report, he was apparently hit in the face with a toy truck, and it was breaking his skin, he was bleeding a little bit. The toy had Nocardia on the surface of it, and the bacteria got in and established infection, and he got a little abscess that formed on his cheek. They successfully diagnosed it. Good job, lab, and good job, provider. I guess difficult to diagnose. They treated him with antibiotics, and he was perfectly fine after that. So, not the usual presentation, and not the usual patient either. The usual patient is usually somebody who's older, hospitalized, has underlying comorbidities, and lower immune function. But this just kind of illustrates the randomness of some of these Nocardiosis infections. Okay, a lot of stuff. Any questions about Mycobacterium or Nocardia? Okay, I will see you guys tomorrow at 10 o'clock.